discovery company, announced positive preliminary results from its Phase 2A trial evaluating ISM001055. The first-in-class drug for treating idiopathic pulmonary fibrosis targeting TNIC showed safety and efficacy, with a dose-dependent response in forced vital capacity, a key lung function measure in IPF patients. IPF is a chronic, age-related scarring lung disease causing progressive lung function decline. Common in individuals over 60 years old and affecting about 5 million people worldwide, IPF has a poor prognosis with a median survival of 3 to 4 years. Current treatments cannot stop or reverse disease progression. Insilico Medicine used the PandaOmics, a proprietary AI-powered target discovery engine, trained on omics and clinical data related to tissue fibrosis. And one novel target, TNIC, was prioritized for further analysis. They then deployed Chemistry 42, a generative AI chemistry engine with 500 predictive models. This engine evaluates molecules for target affinity, stability, and penetrability to generate a desired drug candidate from scratch. Out of 79 synthesized molecules, ISM0055 showed promising results in improving fibrosis and exhibited a good safety profile in bleomycin-induced mouse models. This work was completed in 18 months from target discovery to PCC nomination with a fraction of cost compared to traditional approach. In November 2021, just 9 months after its PCC nomination, our group announced that the first healthy volunteers were dosed in a first-in-human trial of 055 in Australia. Following the success, a Phase 1 trial in New Zealand with 78 volunteers began in February 2022. The study found 055 was safe and well tolerated, with no serious adverse events. A subsequent Phase 1 study in China confirmed these findings. A novel small molecule designed utilizing Insilico's proprietary AI platform has previously been described in the Nature Biotechnology paper published in March 2024, detailing the discovery of TNIC as a novel therapeutic target for IPF. In 2023, Insilico launched 2 double-blind, placebo-controlled Phase 2 clinical trial in both US and China. The Phase 2A trial that has just concluded in China evaluated the safety, tolerability, pharmacokinetics, and preliminary efficacy of 12-week oral 055 dosage in 71 subjects with IPF. Preliminary results from the study show 055 demonstrated a good safety profile and dose-response trend in lung function in IPF patients. We aim to explore extended treatment durations and larger patient cohorts in Phase 2B to further investigate the therapeutic potential of 055 in IPF. Insilico Medicine, a clinical-stage generative AI-driven drug discovery company, announced positive preliminary results from its Phase 2A trial evaluating ISM001055. The first-in-class drug for treating idiopathic pulmonary fibrosis targeting TNIC showed safety and efficacy, with a dose-dependent response in forced vital capacity, a key lung function measure in IPF patients. IPF is a chronic, age-related scarring lung disease causing progressive lung function decline. Common in individuals over 60 years old and affecting about 5 million people worldwide, IPF has a poor prognosis with a median survival of 3 to 4 years. Current treatments cannot stop or reverse disease progression. Insilico Medicine used the PandaOmics, a proprietary AI-powered target discovery engine, trained on omics and clinical data related to tissue fibrosis. And one novel target, TNIC, was prioritized for further analysis. They then deployed Chemistry 42, a generative AI chemistry engine with 500 predictive models. This engine evaluates molecules for target affinity, stability, and penetrability to generate a desired drug candidate from scratch. Out of 79 synthesized molecules, ISM0055 showed promising results in improving fibrosis and exhibited a good safety profile in bleomycin-induced mouse models. This work was completed in 18 months from target discovery to PCC nomination with a fraction of cost compared to traditional approach. In November 2021, just 9 months after its PCC nomination, our group announced that the first healthy volunteers were dosed in a first-in-human trial of 055 in Australia. Following the success, a Phase 1 trial in New Zealand with 78 volunteers began in February 2022. The study found 055 was safe and well tolerated, with no serious adverse events. A subsequent Phase 1 study in China confirmed these findings. A novel small molecule designed utilizing Insilico's proprietary AI platform has previously been described in the Nature Biotechnology paper published in March 2024, detailing the discovery of TNIC as a novel therapeutic target for IPF. In 2023, Insilico launched two double-blind, placebo-controlled Phase 2 clinical trial in both US and China. The Phase 2A trial that has just concluded in China evaluated the safety, tolerability, pharmacokinetics, and preliminary efficacy of 12-week oral 055 dosage in 71 subjects with IPF. Preliminary results from the study show 055 demonstrated a good safety profile and dose-response trend in lung function in IPF patients. We aim to explore extended treatment durations and larger patient cohorts in Phase 2B to further investigate the therapeutic potential of 055 in IPF. Insilico Medicine, a clinical-stage generative AI-driven drug discovery company, announced positive preliminary results from its Phase 2A trial evaluating ISM001055. The first-in-class drug for treating idiopathic pulmonary fibrosis targeting TNIC showed safety and efficacy, with a dose-dependent response in forced vital capacity, a key lung function measure in IPF patients. IPF is a chronic, age-related scarring lung disease causing progressive lung function decline. Common in individuals over 60 years old and affecting about 5 million people worldwide, IPF has a poor prognosis with a median survival of 3 to 4 years. Current treatments cannot stop or reverse disease progression. Insilico Medicine used the Pandaromics, a proprietary AI-powered target discovery engine, trained on omics and clinical data related to tissue fibrosis. And one novel target, TNIC, was prioritized for further analysis. They then deployed Chemistry 42, a generative AI chemistry engine with 500 predictive models. This engine evaluates molecules for target affinity, stability, and penetrability to generate a desired drug candidate from scratch. Out of 79 synthesized molecules, ISM0055 showed promising results in improving fibrosis and exhibited a good safety profile in bleomycin-induced mouse models. This work was completed in 18 months from target discovery to PCC nomination with a fraction of cost compared to traditional approach. In November 2021, just 9 months after its PCC nomination, our group announced that the first healthy volunteers were dosed in a first-in-human trial of 055 in Australia. Following the success, a Phase 1 trial in New Zealand with 78 volunteers began in February 2022. The study found 055 was safe and well tolerated, with no serious adverse events. A subsequent Phase 1 study in China confirmed these findings. A novel small molecule designed utilizing Insilico's proprietary AI platform has previously been described in the Nature Biotechnology paper published in March 2024, detailing the discovery of TNIC as a novel therapeutic target for IPF. In 2023, Insilico launched two double-blind, placebo-controlled Phase 2 clinical trial in both US and China. The Phase 2A trial that has just concluded in China evaluated the safety, tolerability, pharmacokinetics, and preliminary efficacy of 12-week oral 055 dosage in 71 subjects with IPF. Preliminary results from the study show 055 demonstrated a good safety profile and dose-response trend in lung function in IPF patients. We aim to explore extended treatment durations and larger patient cohorts in Phase 2B to further investigate the therapeutic potential of 055 in IPF. Insilico Medicine, a clinical-stage generative AI-driven drug discovery company, announced positive preliminary results from its Phase 2a trial evaluating ISM001055. The first-in-class drug for treating idiopathic pulmonary fibrosis targeting TNIC showed safety and efficacy, with a dose-dependent response in forced vital capacity, a key lung function measure in IPF patients. IPF is a chronic, age-related scarring lung disease causing progressive lung function decline. Common in individuals over 60 years old and affecting about 5 million people worldwide, IPF has a poor prognosis with a median survival of 3 to 4 years. Current treatments cannot stop or reverse disease progression. Insilico Medicine used the PandaOmics, a proprietary AI-powered target discovery engine, trained on omics and clinical data related to tissue fibrosis. And one novel target, TNIC, was prioritized for further analysis. They then deployed Chemistry 42, a generative AI chemistry engine with 500 predictive models. This engine evaluates molecules for target affinity, stability, and penetrability to generate a desired drug candidate from scratch. Out of 79 synthesized molecules, ISM055 showed promising results in improving fibrosis and exhibited a good safety profile in bleomycin-induced mouse models. This work was completed in 18 months from target discovery to PCC nomination with a fraction of cost compared to traditional approach. In November 2021, just 9 months after its PCC nomination, our group announced that the first healthy volunteers were dosed in a first-in-human trial of 055 in Australia. Following the success, a Phase 1 trial in New Zealand with 78 volunteers began in February 2022. The study found 055 was safe and well tolerated, with no serious adverse events. A subsequent Phase 1 study in China confirmed these findings. A novel small molecule designed utilizing Insilico's proprietary AI platform has previously been described in the Nature Biotechnology paper published in March 2024, detailing the discovery of TNIC as a novel therapeutic target for IPF. In 2023, Insilico launched 2 double-blind, placebo-controlled Phase 2 clinical trial in both US and China. The Phase 2a trial that has just concluded in China evaluated the safety, tolerability, pharmacokinetics, and preliminary efficacy of 12-week oral 055 dosage in 71 subjects with IPF. Preliminary results from the study show 055 demonstrated a good safety profile and dose-response trend in lung function in IPF patients. We aim to explore extended treatment durations and larger patient cohorts in Phase 2b to further investigate the therapeutic potential of 055 in IPF. Insilico Medicine, a clinical-stage generative AI-driven drug discovery company, announced positive preliminary results from its Phase 2a trial evaluating ISM001055. The first-in-class drug for treating idiopathic pulmonary fibrosis targeting
right in the office, you can bring it here. There will be a bunch of overflow crowd coming a little bit later. So we're going to have a massive TV outside broadcasting this on LinkedIn. I hope that you can hear me on the other side, and let's also check with our folks. And let's get some more cheers for people who are here, so we don't stand. First of all, big announcement today, so I'm not sure if you've seen what we've announced in the morning. And we didn't really provide media with much of the information. Unfortunately, at this point in time, we have to be quiet, and we have to avoid any significant kind of overly promotional statements, how the clinical trials are. And since we're planning to release the data in a top peer-reviewed journal, also go for conferences, we have to avoid putting the charts in. But what happened today in the morning, we've got our first anti-fibrotic drug phase 2/1A readout. That drug is so dear to my heart because we've started working on that drug only after we developed the end-to-end AI drug discovery and development platform that some of you use and love, and some of you still don't use it and don't love it. So please try to get in there now because it's validated beyond any reasonable doubt. And once in 2019 we managed to get there, we decided to test this platform to run the end-to-end research program. And we even started a documentary video because we thought that we were going to fail. So we identified a novel target, TNIC. Most of you have never heard about this target. Only at that time, maybe 90 to 100 papers published just mentioning the gene. And we designed the novel molecule with our chemistry 42 with a generative AI system. The generative AI system that I kind of proposed 2016, published a few papers. A lot of people were laughing at us, but now they're not laughing, they're using it. And that system allowed us to do multi-parameter optimization to achieve very unique molecular profile by synthesizing only 79 molecules. We took this program through many preclinical studies, resulting in nomination of 3 preclinical candidates. So it's a lot. In IPF, in kidney fibrosis, and IPF inhalable. We also have a lot of data on skin fibrosis, liver fibrosis, but without the official nomination of the preclinical candidate. We started phase 0 in Australian healthy volunteers, 8 healthy volunteers during COVID. Then we did 2 phase 1 studies in New Zealand and in China, and then started 2 phase 2s in New Zealand and in China. And after phase 1s were completed, we published the Nature Biotechnology paper that many of you read. If you haven't, just read this one out of Insilico. Now we publish 1 every 10 days. So it's hard to figure out which paper to read first, but that one, please read that one. It's on TNIC and its efficacy in preclinical and safety in clinical models. And that paper made some noise, but people still were asking, okay, where is phase 2? And now, last week we unblinded, and that unblinding made me cry. Like seriously, Dr. Ren, my co-CEO, called me at 2 a.m. in the morning last Tuesday, and they processed the data over 24 hours after unblinding. And in addition to safety, which we expected to see, we kind of when you're running the clinical trial, and if you don't see any surprises, usually it should be reasonably safe. But the unblinding also showed dose-dependent improvements in forced vital capacity, the critical biomarker in IPF patients. So usually on standard of care, you always see the decline. Very rarely you see any improvement, and you are trying to slow it down. Here, you're actually seeing improvements in forced vital capacity. And again, unfortunately, I'm going to show you the pictures right now, but to me, they were thrilling. And I really cried, seriously. So today in the morning, we have made this announcement. We're going to celebrate later. We're always celebrating later, sometimes never. You have to, show must go on. And this is a huge achievement for the industry because so far I do not know of any other company that have discovered a target using AI, their own AI, generated the molecule using their own AI, and took it all the way to phase 2A complete with efficacy. We did not expect efficacy at 12 weeks, honestly. So huge positive surprise. Hope to publish it in a good peer-reviewed journal. So if you know anybody at NEGM or Lancet, please give them a call. And we're now talking, so it's kind of like scientific business development. Your guys are TD. Our job is to give you the pipeline assets, and we are now selling it to the journals. But speaking of the pipeline assets, so most of you may not know what Insilico is. Most of you probably do. So we sell software. We put software on the market. We have 3 platforms: biology 42, chemistry 42, medicine 42, and science 42. Biology 42 for target discovery, chemistry 42 for small molecule chemistry, medicine 42 for clinical trial analysis, and science 42 for all things science. We're also doing now carbon capture. We're doing hydrogen storage, and many other very interesting explorations around all things science. As a matter of fact, once your generative AI software performs really well in agriculture and you realize that your molecules are safe for bees and other animals, it makes your overall generative reasoning system better, because chemistry is chemistry, especially if it's one model. Helps you go across all of disciplines. We recently launched Dora. Dora is like super cool tool. It allows you to write research papers from scratch. So if you are doing a PhD, or well, if you want to, so what it does, it basically sprouts a bunch of agents. So some of the agents are PIs, some of the agents are workers. They perform valuable research and then bring you a paper together with original references that you can like or dislike. So we actually we don't use your data, we don't see your data just like with other tools, but we get your feedback. And the more feedback you get, the better our tool becomes for the entire community. And that community is improving all of our tools. So the more you use it, the more validated the system becomes, and then we can utilize it for massive explosion in drug discovery programs. But the most important part that I believe is making a huge difference is that we managed to develop a therapeutic pipeline. And we are not planning to take it all the way to the market. At least I don't see myself doing that yet. Our job is to fill the pipelines of pharmaceutical companies with ultra high quality assets. That's why the quality of the data room that we usually have for the drug exceeds the internal standards of the pharma company. So we try to go above and beyond what you got to use to internally and also from other biotechs. So when you open up the data room, we really want to buy. And the quality of those drugs is very high. So we are a factory of drugs. The faster you buy the drug from me, the faster I can create 2 or 3 more. My limit is now capital, right? So we actually think that we can do 12 preclinical candidates every year. 2022, we did 9. And 9 have reached clinical stage. Show me another AI drug discovery company that can do that. Any drug discovery company. And of course, one phase 1A readout is truly amazing, and I'm very bullish on this drug. No promotional purposes, but I even designed a really specific shape and color for the phase 2B trial. So we're ready. And those assets are usually designed so that we're trying to test our target discovery system and go for a high level of novelty, like with clinic, or we're trying to go after what you want, but not now, 3 years from now. So our job is to predict what the pharmaceutical companies want. And we want you to buy those assets later to combine it with your pipeline. And we think that the pharmaceutical companies are vastly superior than us at running combination clinical trials, running multi-center clinical trials. You get more trust from the patients, you get more trust from the clinical centers because of the brands, right? So patients are more likely to take your drug than mine. Hopefully not anymore, but we'll see. And we really want to partner with you to fill your clinical stage pipelines. And you should be pretty good at AI internally for running clinical trials. So our main job now is to sell and license the drugs to you with ultra high quality so you can develop them. And for that, we have business development. So let me present to you Michelle Chan. She can talk a little bit about her background, but she is a very seasoned, experienced business professional who's worked for the company for 3 years. And you've seen some of our deals like USP1 license to Exelixis, $80 million cash up front, very, very substantial deal at phase one. Menarini, $12 million cash up front, $500 million total deal value. And now some of these are in the term sheets, so you should be able to see some more announcements, hopefully, right, Michelle?
That's the hope.
Yeah. So we should be doing more and more deal making. But we want to ask you, what do you really want in a partnership? How do you see the partnership happening? And for those of you who are interested in partnering with pharma or establishing, let's say, massive AI data centers and data capabilities to see what pharma wants, this is a great panel. And we're also live broadcasting on my LinkedIn. Somehow I became a little bit of an influencer there, so a lot of followers. And I really like to post, so and I try not to post personal stuff. I'm not Elon. Sometimes those comments are a little rough and over the edges, so let me know. But now we're also doing live broadcasting and training myself how to do that. Sometimes you will see myself doing that from the Middle East, from Saudi, from Southeast Asia, and sometimes somewhere from the mountain where we are collecting specimens for high throughput screening, and sometimes from the lab. So welcome to our BD panel. Michelle, please, the show is yours. We have the wireless mic here, so we have to kind of go around. And this one is a second wireless mic that we're going to be using for the panel.
All right. Okay. Well, as Alex mentioned, welcome to Insilico Medicine, our Cambridge office. We're so excited and honored to have you here. So I'm very privileged to have my very esteemed panelists join me. So maybe you guys can come over. I will sit here as the moderator for the panel. Monica, you can sit here. Claire, Dave, Home, and Natalia, here the cookies, the crackers. You can see here. So we are, the theme of this panel, we're talking about pharma business development and the deal making. I think what I would like to do is have my panelists introduce themselves a little bit, say a couple words about their company, and then we can dive into Q&A questions. So start with Monica.
Hello, it's great to be here. Thank you very much for the invitation. Pleasure to be here because Insilico is our partner. I'm Monica from Sanofi. I head our partnering and business development. That means BD deals, that means alliance management, so we stay very close to Insilico, as well as our venture investments. And Sanofi is a big pharmaceutical company across therapeutic areas, and
Claire.
Yep. Hello, everybody. I'm Claire Fisher. I'm the SVP of Business Development and M&A at BeiGene. We are a global oncology-focused biotech company, and BD for us is actually quite similar. So we cover everything, soup to nuts in business development, as Monica described, including venture. We are quite unique. So we're pretty vertically integrated. So we have about 1,000 research people of our own, about 3,000 clinical developers. And so our pipeline is quite internally generated, but we're looking a lot of the time at companies like that. So excited to be here today and meet everybody.
Hi everybody, I'm James. Thanks so much for the invite to come and meet everybody today. I lead research and external innovation at Ipsen. We're a mid-sized global pharmaceutical company with our roots in France over 90 years. ago, and we're in 3 therapeutic areas: oncology, rare disease, and neuroscience, with a mix of marketed assets all the way through to basic research. The approach that we take to business development and growing the pipeline is a little bit different to what we've just described. We have a near enough exclusive focus on external innovation and bringing in assets from the outside to then focus on developing them. And at this point, we're about a 5,000 person company, about 3 billion in global revenues across the split bit evenly between the US, Europe, and the rest of the world. And we're always interested in new partnering opportunities, and we're delighted to be here today.
Hello, my name is Hong Chen. Thanks for the invitation. I work at Novo office. I'm the director of search and evaluation, looking for external opportunities for Novo. And as you know, Novo has been very successful recently with obesity drug. The company is also very famous for the insulin. So if you don't remember, at least in our signal, so far the Novo has been a two peptide company, which has been existing for 100 years. Now, of course, we're looking beyond the peptides and looking for other modalities, and we're looking for the opportunities to supply the pipeline. The other thing I want to mention is Novo now has a big presence in Boston. We started a few years ago, only like 2 or 3 years ago. Now we have a very significant presence both in BD and also in research. Looking forward to talk to all of you.
Hello, everyone. Thanks, Michelle, and for inviting me. It's a pleasure to join this panel. So I'm Natalia Mianova, Director of Business Development for biopharma. So I'm focused on leading search and evaluation efforts in oncology. So Astel is a primarily oncology company. We are the company is headquarters and origin from Japan, but we are a global pharmaceutical company present in over 90 countries. Mostly oncology, so branded products in Insilico, prostate cancer is our biggest drug. We also have Padcev. So those are partner programs. So we have actually a bigger alliance management growth because of how many partnerships we have. So we partnered with Insilico on software, and hopefully we can also find ways to partner on therapeutic areas. In oncology, though, which is our major research activity, we've been very strong on small molecules. We now created a group called Engineered Small Molecules that is focused on protein degradation approach, molecular glues, protein degraders, and such. So mostly this focus on oncology.
Yes, we'll just go ahead and grab the phone right here. I think since you guys are business development experts, so one of the questions I have for you, if you look at your internal pipeline, right? I'm curious about what percentage actually utilizing from external sources, and then also very interesting the number, like how many opportunities we evaluate per year, and what percent of these two are real deal. Just give us some at a high level the volume and how much work people appreciate on the buy side that the things you have said through.
Sure, so partnering is very important in Sanofi. And I didn't tell you what therapeutic areas we're interested in, but I'll tell you a little bit about the pipeline. And in that, I'll highlight what we're interested in. Our late stage pipeline, we have big 12 assets that are either going through phase 2 or phase 3. All of them have a $2 billion plus potential. So we call them our big 12 blockbuster potential assets. A lot of them are in I&I. That's the most important area of interest to us. And there we will partner across the stage of development, from very early to late. To answer your question, our big 12 pipeline is all partnered, and we're proud of it. So those molecules came through either licensing deals, collaborations, or M&A. Overall, our pipeline is about industry standard, about 50-50, organically developed and partner. But the ones that we're really focused on that is going to secure our growth throughout the 2030s and beyond, those came from partnering. And partnering is a lot of work. We evaluate over 200 opportunities a year, In venture, we evaluate 1,000 opportunities a year. And we do about 20 licensing deals, a handful of M&A, and about 8 to 10 venture investments a year.
Most of our later stage pipeline is probably organically homegrown pipeline, not entirely, but mostly. And a lot of that's sort of more human focused. This year, we're actually putting 12 solid tumor products into the clinic by the end of the year, so quite a way through that, but not yet quite complete. Again, the majority of those are actually being generated from our internal research programs. But I think, as everyone's aware, oncology is really about combinations. So we're really very actively seeking external partnerships for mechanisms of action and asset areas that we haven't developed internally to really complement all of that current pipeline. So very busy for us too. I think we probably I'm looking at Christina sitting in the front row because she does a lot of this hands-on, but we look at hundreds of things a year as well. And mostly in the oncology space because that's our primary focus. We look a little bit at I&I, if it's something that we think we could really use our capabilities and our expertise to really move forward very effectively. But hundreds of opportunities every year, and I think it really varies from year to year, how much results in a deal, because sometimes it might be for us that we do less but bigger deals, or we could do more but smaller deals. So it does really change.
Is there a mandate for deal? Like you to do 2 or 3 deals a year? So it's really depends.
We don't tend to mandate it. I mean, we have some guidelines around what we'd like to achieve, but assets are assets, right? And sometimes there's a wealth of interesting things to look at, and sometimes it's a little bit less. So we're not too rigid about it, and we really want to make sure we're effectively bringing in the external innovation that we need.
Right. So as I think I mentioned, we have a slightly different business model at Ipsen. We don't have a competing internal pipeline. Everything we do is through partnering. That's the business model that we adopted 5 or 6 years ago now, and we've been using that to transform the overall company and also the pipeline. Back in 2020, we had 1 molecule that was selling more than next year we're projecting to 4, and going into 27 we're looking at 7 or more. So we're very much further grow the pipe across all stages. We're always very interested in what's available in the later stages of development, the registration phase, and we always look to think about how we as a mid-sized company can leverage the capabilities that we have to partner in a slightly different way than perhaps you might see a large pharma company doing. From a capability standpoint, we keep everything you would expect a pharma company to have in-house to do basic research, all of the clinical development, the preclinical development, global regulatory, marketing, etc. But we look to really source the innovation through our partners. We're very proud that we have a long history of several outstanding partners, and we're looking to continue to add to that. From a therapeutic standpoint, we're probably about a 60 or 70% oncology focused business at the moment, but we have a long history in neuroscience through the toxin franchise, and also through rare disease, where we've been very focused recently on various rare liver diseases and building a suite of assets and a franchise around that we're looking to continue to complement.
I think our role is kind of different for most of the big pharmas. We're not extremely focused. So after 100 years, we were mainly focused on diabetes and now obesity, and also slowly we're expanding to cardiovascular disease. We also have a separate rare disease division. That's about it. But on the other hand, we are very actively investing in technology, like enabling technology, like I said. We want to go beyond peptides, antibodies, small molecule nucleic acids. So from disease area, I think we feel very confident about our, obesity treatment and the diabetes. So we're really looking for transformative new innovations. In that way, we actually can cure this disease, not just control them. So we're really looking for new ideas and new innovations, both for technology and for action. So as for how many fields, I only joined Novo, I think, almost 3 years. I just counted my own Excel sheet. I'm not AI yet. I'm Excel. Excel. I already have like components 300. Yeah, just for myself. I'm not even talking about my colleagues. So you do have to look into a lot of opportunities, and for the deals made, I think every year by the people in the council. What I think is very unique for, at least from our cases, it takes long time. Some of the deals like one recently I'm bringing to our transaction is actually I started it more than 2 years ago. But we are very patient. We are old company and we have been here for a long time, so we look at long term. So we don't look for the quick, if something really have potential. We work together. For 1 year, 2 years, just it makes sense. We just keep going. So I think good thing for us is to keep good relationship and get to know everyone and then working together. So I think the best beneficial for both sides versus just one big country. So the other last thing I want to mention that is for how much innovation. So insulin and the semaglutide, those are homegrown for us. But like our cardiovascular pipeline is completely external. So we're looking more and more externally for more and more collaboration.
So on Astellas, as I mentioned already, it's very big on partnering. In general, so a lot of our research programs are partner, but also we grow internal programs through acquisitions. We acquired quite a few biotech companies in the US recently. So we have a total of 11 affiliates across the globe. And so it is all in a sense of partnerships. And in the current focus is a little bit a step away from just strictly therapeutic area focus, but also a big focus on modalities. So we bring novel platforms to create programs together, so research collaborations. And some examples maybe on oncology is another one. Oncology, we brought catalytic virus, cell therapy, several cell therapy partnerships, and different IPC, in vivo CAR, allogeneic partnerships, so all of them there exist there, and including also T-cell engagers and mass technologies and atomic partnerships. So a lot of those partners.
Great. So, I guess my third question is that, you heard from Alex talk where a platform company or wonderful pharma AI technologies available. We also have assets, we should number here. I guess from buy side point of view, what's your view on platforms versus assets? And as an AI company, and I have to ask this question, please be honest with us. And is AI hype? Is it real? Your personal opinion.
I'll start with the second one. AI is very big at Sanofi. We call ourselves the tech-powered pharma. We use AI internally. We all have an app on our phones that tells us in real time about the key financial metrics for the company daily. So if I want to see what our sales are year to date, month to date, and today across the main businesses, I have it on my phone. I have the P&Ls. I have our pipeline on the phone. I have the main milestones of each of the big 12 I mentioned. So I think AI is here to stay, and it's going to be in our industry, it's going to be across society. Pharma and healthcare generally has been kind of slow adopters of a lot of technologies. I think we're changing that with AI. And it has proven itself through partnerships like this one in drug discovery. I think the next area where we're focusing on how we can deploy AI better is in designing clinical trials and in clinical development. How can we accelerate clinical development with AI? And we have a few partnerships there as well. So I think AI is very important, and it will we don't know what we don't know yet. So we're all learning, and we'll see how it continues to be applied in our industry and in society, as I said. And your first question was?
Platform versus
Platform.
assets, yes.
I think it's a little cyclical. You know, I've been a deal maker in this industry for over 20 years. When I joined in early 2000s, it was just the end of the platform boom, genomics. And then we just saw that kind of the same cycle in the 2020s. A lot of public companies that were just platforms went public. I think we're coming back to assets. Sanofi did a lot of deals in late 2019, early 20s for platforms. Right now, we want to focus on assets.
Rather than something more tangible.
Yeah, maybe picking up from that last point first. So I totally agree. It's very cyclical. I think ADCs are a pretty good example of the activity of the
You can spell the word,
Yeah.
you can spell it.
And now people are really looking for the proof of the different platforms and the assets. And I think we definitely work in that area too. Platforms are very important to us. There are certain areas that we very much want to stay the leader in from a platform perspective. But I think most of our activity is probably in assets at this point in time, throughout the spectrum of early to late. On the AI side, I think, as Monica said, it is popping up everywhere. We are also very active in developing expertise in this space. We also focus probably for the moment a little bit more on the clinical side, how we can use it in clinical design, development, regulatory, are all very much active areas for us. I think on the research side, we're staying very close to it. We're trying to monitor it all as close as we can. At the end of the day, I think for us, it's the targets that matter. So sort of seeing what the output is, but staying very close and definitely recognize that it's going to be a big piece of what we will do going forward.
Well, firstly, I'm certainly not going to go back to my CEO and CFO after this meeting and telling them that they can get an app on their phone that's going to give them a lot more complicated. So, I think for us, given the business model, we're more focused on the asset side of things than the platforms. I think for us, it's really about the target, the strength of the science, how's it going to fit into the space. For the earlier ones, how are you going to get into clinical proof of concept? What's that line of sight to registration? So I think, we very much look for AI for how we can apply it to expedite our clinical development studies and the other things, but also very much look to potential partners for how you can apply it to the drug discovery process, because there's a lot of good evidence. Yes, it's a great example of it, of how it can really help to reduce cycle times, integrating all of the data that, yes, we can do with people, but to do that more rapidly. I think there's some really good examples now of molecules that are coming through the development candidate stage through into the clinic that show how you can really practically apply it. But ultimately, you've got to get the targets right, because if modulating the target doesn't matter so much in human disease, then it doesn't matter how you arrive at the molecule that you get to. It's really about the clinical activity and the balance with safety that really matters.
So I think from our perspective, it's slightly different. Like I keep saying, our biggest drugs are peptides, but that is supported by extremely important technology platform. It is successful because the target is well validated for 100 years, but it's a technology that brought out the real drug. So we're extremely focused very much on the platform, but it's not just in general, but specifically. For example, tissue specific delivery is extremely hot. It's as important as assets. The other one is, for example, like sustained drug release. Because now, you have injectable once weekly, everybody's happy, but how about once monthly? How about every half year and 1 year? So if you have that kind of technology, time will be very, very interesting. Or oral, right? Like peptides, you have to inject, but if you can make it oral, and then if you can improve the bioavailability. Yeah, so that's how we look at the technology is what we need versus whether. So for the assets, we're very active looking for assets, like I said, but on the other hand, there are very few new ones yet beyond what we have built one, because many of the companies do work one now, but what beyond that? So looking for new things is what we're aiming for. For AI, I just actually was at kind of panel on LS conference. Michelle and I were there. Somebody asked the same question, so how do we evaluate? I think in general, everybody knows that AI is important. But for us, and the search evaluation was the gatekeepers to bring in these things, what I really need is some kind of example of success. Like Alex just said, look at my AI and then phase 2 success. Then that's more than enough. You don't even have to show me what box, this box, that box, something come out. I don't need that. I don't understand anyway. But you just show me, this AI bring you the success. I think that's the first step. I don't mean the real evaluation. This is just how I can understand it, can feel confident that I can introduce to my colleagues.
Yeah, what you said resonates with me also. I was also on the list sec. So I'll answer the second question first. I got a lot of panels were not about AI, but it came up. So there's definitely hype, but different people mean different things. So you have to. It definitely, in Astones, we use AI already for drug discovery. So some other applications we are looking into it. Definitely want to optimize and all, but there's definitely a hype element because there are probably not that many areas where there's a large data set that we need AI or could benefit from. So there's certain areas. So that's the first question, and the second one. The first one was asset versus platforms, right? So same as one, where we already made large investments and are interested in, for instance, in addition to oncology, we are investing in gene therapies. So there we use the ADD approach. So we've invested already in manufacturing capability. We need assets, but also we need platforms to improve that sort of thing, gene therapy approach. So we are doing partnerships in these areas. And but definitely assets is something we need more because our pipeline is growing now. But we invested in different modalities. We have a lot of preclinical internal partners in internal pipeline. But assets we need more in the phase 2 stage. So I think it's great success today to see our phase 2 asset becomes interesting. Similar to what you said, we're also looking at let's see if the AI fully implemented. Let's see if it's actually successful to bring a successful clinical stage.
Well, I'm going to ask my last question because I open up for the audience here. So I want to talk about deal making. So think about experience you have, some good, some are bad. Maybe just pick just one, either good or bad, just pick one. Hopefully we'll have good balance here. And talk about that particular example, what makes really good, what makes really bad. And maybe you can also give some kind of advice for a company like Insilico Medicine, as you hear from Alex, we're 100% about collaborating and want to become a discovery engine to pharma, and maybe some things we need to watch out for so that we can learn from and become better.
So maybe in the interest of time, rather than a specific example, maybe just a few things that do make for a smooth deal. One, investing the time and building the relationship. So deals take a long time. Invest up front, and maybe even years before you do a deal. But as your technology is maturing, as the asset is maturing, continue talking to the partner, update them on the science, and build a relationship. And the other thing is know your audience. So if you're talking to a scientist about data, or if you're talking with a business person, connected to the business and talk about how is this technology or asset going to fit with the company. So maybe those two things.
Next one.
Yes, I was going to go the general route as well. So I would say, to complement Monica's points, be clear about what you want, communicate clearly what you want, so you're not wasting time dancing around the structure that you really ultimately would like to achieve. And with that, be a little realistic about your own capabilities, I think. So if you want to partner, collaborate rather than a license, be clear about what you're bringing to the table and what you can really bring to the table, so that you're not going to fall short on resources and jeopardize the whole relationship halfway through your first clinical study or something like that. And be data driven. So by that I don't just mean with scientific data, but when you're coming to a discussion or a negotiation, be data driven, bring precedents, bring points of good information that can help the other side really understand what you're looking to achieve as well. I think it's very helpful when you mention point to examples. So they're the 2 or 3 things that I've mentioned.
Yeah, building on that, I think from our experience, transparency and honesty are really, really important. So that, on the buy side, we know that all the biotech companies are super excited about their data, and that's sort of the job of be realistic about what you've got, how it could fit, how it might translate. Because I think the worst experience is everybody gets excited, and you go into a data room, and then you peel peek behind the green curtain. It's like, oh, you think you're looking at a Ferrari, and it's actually a lawnmower. So I think that is really, really important. Really, really important. And I think also, for us, we really like to have the conversations where both sides bring something to the table, so the relationship becomes more than just about the financial aspects. So again, we love the conversation of we really like to do this, we think we can deliver it, this is why we think we can deliver it, how would that fit, so we can really start to craft each deal in a unique way. Those are things that really resonate with us when we're having the conversations.
I agree with all of you on what you have said, and just one more point. I think for Novo, even though it's now number 2 based on the market cap, but the company mentality actually is smaller, much smaller than that. I feel like it's a middle-sized company. So when you approach us, you just feel like you're talking to equals. We don't have a lot of process. You have to reach this, that. It's all a personal relationship. We talk together. We openly discuss what we want, what you want, and then we figure out how to get there together. And that's the kind of experience I have had so far.
Yeah, a lot of good things were really said. So I wanted to maybe, as you said, a bit honesty, transparency. There's also common end goal, right? Because sometimes some of these assets go different directions. So there may be that you have one view and the partner a completely different one. So this is long relationship through clinical development. You stay. I mean, assuming this is for development or commercialization, not just about licensing, you would be involved in long years of discussions, and especially areas where you may it may not be relevant to this audience, but if you do a kind of regional split and start to kind of be less transparent with your regulatory discussions, where problems could arise, because ultimately all of the patient data is shared and all that. So all of this decision making, kind of JRC meeting, governance process have to be very well thought through, agreed by most parties, and the real kind of end outcome of what we want this collaboration to look like has to be well aligned.
Thank you. Any question from the audience? This is the rare opportunity you can ask the executives.
I have a question.
Yes, go ahead.
I can use the mic.
Maybe you can introduce yourself.
I will be the mic man.
Sure. Perfect. Thanks, Alex. My name is Ray Dogo. I'm with Bio. So I guess my question is, when you're working and collaborating with AI companies, what level of feedback do you provide in terms of like reinforcement learning? So are you clicking I like this response like on a daily basis in terms of, if you're generating a certain report or something, are you manually every day kind of reinforcing the learning process at the company, or does that question make sense? Are you, no?
Well, I guess the question is, how do you provide feedback, right?
How do you provide feedback maybe within the software itself? If you're interacting with the AI software directly, are you providing feedback on the software? For example, with ChatGPT, you can like a response, which reinforces the model. And I'm just wondering if that's the level that business development people at pharmaceutical companies are doing at this point in time.
I can answer this question. They don't.
No.
Right now, like,
is that a vision for the future?
Well, we have right now like 10 out of the top 20 pharma companies use our software. You cannot provide feedback easily. You cannot get their feedback easily. Like or dislike, it's all automatically embedded in the software. And actually, what you are interested in is a massive dislike. If they kind of bet, let's say, 100K on a program that was generated out of your software, and specifically a specific model, and they tell you that it failed, well, great. Well, you might have destroyed the relationship there, but it's better to explain like why. And while you're explaining why, you're improving the software. So that's and then you make it available for the community. So plug a new model. So that's one. But of course, that's why it's a good idea to deploy lighter software like Dora or Pandomics. There they can actually like or dislike, and academics love to do that.
Right.
So that's the job for academics. Pharma doesn't like to do that.
Yeah, appreciate that.
Sorry, just had to step in.
Any other urgent question?
Yeah.
Okay, one more.
All right, so I'll give you my microphone, and it would be great if you can introduce yourself for a second.
Hi, my name is Suman. I'm in the Cambridge Innovation Center in the innovation studio there. My quick question is, do you apply or do you have any differentiated process when you evaluate an AI discovery, AI generated candidate versus other methods? Like is everything going through the same process as far as you're concerned? Yeah.
All right, so we will need to circulate the mic. And guys, let's make it the last question, because later it's so tough questions are best asked at the reception, right?
Softball.
So, very quickly, it is very challenging to evaluate AI. So we don't have in pharma so many. We like to have experienced physicists, PhD physicists working by bioinformatics group who can tell good AI from not good AI right away. So basically, it's similar to even other companies. So it's very hard to say. We are not used to evaluate this sense of. Because we are not looking at algorithm, we look at the end result, which is asset or proposal to hypothesis as such. So we are not able to evaluate algorithms. None of us, and very few people in pharma have such experience.
Anyone else want to comment?
The same comment. We look at what pops out the end, right? So, yeah.
All right, so big hand to our panelists. By the way, so maybe we can take a group picture because you're all female panel almost, right? So we have to introduce some diversity. Next one is going to be all right. So let's do a panel picture, right? But maybe you can stand, and then we will fight the panelists one more time. So this is the group.
Right.
Okay, so just look right at me, and one more good smile from the camera. Awesome, thank you.
Yeah, John, thanks so much. Let me introduce the agenda. So we actually have one more panel, like back to back right away. We have the reception area where we are live streaming this, so you can actually, if we spot, you can actually go there and enjoy the food and drinks. And by the way, feel free to get some tap food and maybe some drinks. I can serve if you like. I have kind of the Swiss Army knife. Today we're going to have the next panel is going to be extremely important. It's probably the most important topic in my life because everything I do is focused on aging research. I think that this is the most fundamental, important biological process in your life because it's 100% certain to kill you, right? And you want it to happen like later and preferably in a healthier state. And preferably maybe even very far in the future. But that process will take everything away from you. And ideally, we need to start developing therapeutics that target this core process in aging. And our second panel is going to be an all-male panel. I apologize for that. You've seen we just had an all-female panel. It's not going to be focused on BD. And our speakers today are the phenomenal two gentlemen who I know for many years. Both of them have made huge contributions to the Boston biotechnology community. So one of them you probably know extremely well, Antonio Regalado. He is the biotech editor from IT Technology Review, and he covers biotech, but also crazy stuff. So crazy stuff like, reprogramming, cloning, brain transplant, whatever. So crazy stuff around the world will be covered by MIT Technology Review first. And sometimes even I'm like thinking, wow, that's literally masterpieces. So please take your spot on the panel. Great, absolutely amazing space. And our second speaker today is Professor Vadim Gladyshev. We've been friends for I don't know how long. We know each other for a very long time. He's made fundamental contributions to the very basic biology of science, of aging research. You probably know ARDD, Aging Research and Drug Discovery Meeting. So that's the largest one now in aging research, probably bringing together biotech, pharma, and top academics. So when we introduce Vadim at this meeting, and he is there from the very beginning, thank you. You made the meeting shine. He doesn't have any conflicts of interest. So this is like ultimate pure play academic researcher who supports the biotechnology community, but he doesn't have conflicts of interest that direct his voice into a specific direction, right? So you can ask him very, very fundamental questions, and he'll answer them with academic honesty. Also, if you get him as a reviewer, be prepared for like very rigorous process. Because he has seen everything. And what Vadim is working on is understanding what is aging, right? So we actually don't know, right? And some of the papers that he publishes are so fundamental that it makes you question life. But today what we want to do is, so Vadim, please take your spot. I hope I didn't spend too much time on the introductions, and I could just go through your credentials, and then your H index is bigger than the circumference of the Earth, right? So huge numbers of citations. So today, what I want to do is try to link aging research and biopharmaceutical industry a little bit, right? And talk a little bit about dual-purpose therapeutics, targeting the fundamental pathways of aging, and at the same time disease, and trying to go to intervene in aging directly. So I just know that Antonio has followed this industry for a very long time. I don't think there is a researcher you don't know, and you've seen probably every, sorry for saying, a DS company out there as well, right? Some of them collapsed dramatically. Very few of them succeeded ever. Actually, I don't think any succeeded so far. And it would be great to chat about some of those experiences. I am going to be moderating, so you're kind of on the panel.
Okay.
All right, great. And I'm gonna sit here. And then we will actually hopefully have a vibrant discussion with the audience, because who doesn't care about aging? Actually, a lot of people don't care about it. And my first question to the panelists is, how do you see aging research propagating into the biopharmaceutical industry right now? Did it become more credible? Is it at the same level over the past, let's say, 5 years? And how is pharma and biopharma addressing aging as a business opportunity?
Tough question. I really can't speak for the pharmaceutical industry. I mean, I don't see any really interest at all. The news that we've written about is usually the entry of sort of new entrants appear, like Altos Labs, $3 billion, but is it a company or is it an academic institute? Evolution Foundation out of Saudi Arabia. I mean, this is still a domain where you have to give away money. And, but there are new people coming in, also the cryptocurrency, billionaires who seem to have an interest in longevity. It somehow fits with their philosophy of life, to beat the rules, I guess. So really, just in terms of the news cycle, it's about new entrants coming in to try and solve aging. Although they're not really new, because 15 years ago we had the founders of Google come in and start Calico. So it's always the wealthy patron who is getting older, and now is the time to cure aging. So that's sort of the phenomenon, I would say.
Yeah, I view aging research and the relationship with pharma, I would say, is glass half full. On the one hand, there's been dramatic progress recently. It's the tools available, I think most importantly, biomarkers of aging. But finally, we are able to quantify this process to some degree, at the organismal level, cellular level, organ level. On the other hand, we still don't understand the nature of aging. There is no consensus on the nature of aging. Just as an example, we gave a questionnaire to experts in the field at a major conference, and they asked, to define the aging process, what causes aging, when does aging begin? There's no consensus on any of these questions. So everybody, but then we ask, do you think you understand what aging is? And everybody understands, but it's just understanding is different, and it's incompatible among various kind of paradigms. So this fundamental basis for the whole industry needs more development, needs development. So these biomarkers we're able to quantify. This means that it should be possible to also develop targets against this process and test these targets, but it's going to be in the future. Maybe we currently don't have a single intervention that works against human aging.
Well, I will, as a moderator, also add to this a little bit and say that some of the pharmaceutical companies actually have prioritized aging research as a platform for drug discovery. One of them was pretty open at ARDD, LA Lilly. They basically said, okay, aging is basically part of our corporate strategy now. We are addressing biological processes in addition to obesity and dementia. We are going into that direction. Novartis has added a huge number of aging researchers led by Colin Wall from ETH Zurich, who is our very close friend and alumni of ARDD. And now this industry is becoming more credible. And also some biotech companies, like, for example, BioAge, which is going IPO. So they are not allowed to, they would have been here, actually, if they were not in a quiet period. Kristen is a wonderful speaker. So they also identified some targets that may be implicated in aging, utilizing their own target discovery approach. In-licensed a few drugs from pharma, and now are purposing those drugs for age-associated diseases and types of biology like muscle wasting, for example, so regaining muscle. Partnership with Eli Lilly, and they're going public, so look them up. I'll probably buy a few shares just out of compassion and belief. Of course, what we are doing with Insilico, many of those targets that you see on the target list, they are dual purpose. So TNIC we've identified using aging research. But I agree with Vadim that we currently don't understand the fundamental biology of aging. AI hopefully will help us solve this problem. And I think that this problem needs to be solved in order for us to have the time to solve everything else. But my question also to Antonio, so you see a lot of flashy headlines.
Mhm.
Some of those headlines are mind-blowing. And some of the media is, they amplify those headlines. Some of them, they actually try to go dig deeper and become more critical. How much substance do you usually see behind those headlines? And
What is the headline that you're referring to? We're not specific about that.
Well, for example, cellular reprogramming.
Right.
Right. So somebody extended life of an animal using cellular reprogramming and told you that it's like 25% and they are in a peer-reviewed journal or it already came out to the peer-reviewed journal. So how much of that usually gets into the...
Okay, well, I'm the author of some of these headlines.
Yeah.
I'll give you the example. We wrote the first story about Altos Labs, which was an interesting journey for me because I had to kind of figure out what was going on in the story. Headline was something like Silicon Valley's latest plan on living forever, defeating death. And then we had a very hilarious picture where we had a scene from the Swedish movie about death. Anyway, what's it called? The Seventh Sea. Right. And the knight is playing a figure, chess, and it's Jeff Bezos. So people were outraged by this headline. I mean, they got all fussy about the headline, including Altos. They were very upset by the headline. They're saying, "Oh, we're not trying to defeat death. We're not trying to defeat aging. We're about health span." I was like, really?
Yeah.
So it was quite interesting the reaction of just of the formulation of the story, which again was billionaires trying to defeat death, because I think they felt that it sort of minimized the importance of what they were doing. They're embarrassed. I don't know if that answered your question. That was a story about a headline.
What about Calico?
Oh, Calico. When Calico became public, that was through an exclusive given to Time magazine, and it said Google's plan to defeat death.
Yeah.
And I think in response, I didn't get the story, so I was upset. So I wrote Google's plan to defeat death, LOL, and I explained why this would actually never work, right? If you're second in the news cycle, you don't have to. The first person has to sort of hype, but the opportunity for the second person is to cut it off at the knees. This is not going to work. And actually, that prediction, of course, was better. It hasn't worked yet, although they continue to do their work.
I can comment. I actually think it's a good idea to organize these companies. Calico, I'm not sure because we don't know much, but at least Altos, they recruited really good people.
Mm-hmm.
So even though it's a secretive endeavor and we don't know what will happen, but these people do some work and hopefully this will advance our science. So because basically it's an additional funding to do basic science, but not only basic science. So not only these companies, but there are, as Alex mentioned, there's maybe Insilico itself is an amazing company by age. There's many interesting companies that hopefully will advance and help us advance because it's a merger of biotech and academic research in a way, because they do basic research. So I'm for it, even though I agree that we have to be skeptical from the academic perspective. With we need to see what's the outcome of that.
Well, my question to Vadim, and get ready. So some of your neighbors at 77 Louis Pasteur, David Sinclair, George Church, they founded companies before, right? Co-founded. Nowadays, it's pretty much like spinning those companies like hotcakes. Every postdoc started something, and they co-founded it, and you remain the purist. So in that building, I think you're the only one who hasn't started a company. My question is why? And second, are we going to see a company anytime soon?
I'm waiting for the opportunity. So, but honestly, I don't want to fail. So basically, I think it will happen. I hope it will happen when there's the right time.
So you're not against companies, you're
I'm
just
not-
waiting for the right opportunity.
That's right. I'm not against
Okay.
companies, but you're right. Sometimes I feel maybe I'm the only in the field. Like I started a company.
It's not a bad thing, it means that
Yeah.
you're actually not conflicted as well. So if Vadim starts something, please break it as a news.
Well, usually I find out by reading the conflict of interest disclosure at the end of the paper. You know what I mean? Like the company name will appear first there, even if it's stealth, the company name appears there. So I'm the person that's racing to the end of the paper to see if he started the company. And actually, most of the companies, the most common type of company to start in the particular space that he works in, the biomarkers, is a company that will tell you your supposed biological age. Like there's many of these companies. You spit, I guess, in the tube, and then they measure the epigenetic status of your cells. So there's professors at Harvard and elsewhere that have a company that will tell you your epigenetic age. Now, is this useful information? I'm not sure. So the quality of those companies, I would say, is in question.
Yeah, well, nowadays I think biomarkers of aging, as Vadim said, that's the most exciting and vibrant industry. So people started selling those tests because they also need to figure out how to make money in aging. Unfortunately, yes. So you get a specific age, what are you gonna do? How are you gonna modulate that? But now we have the tools to track it, and that's great. We just recently showed that you can identify protein targets out of methylation aging clones, which is great. Hopefully, it must work. But without a biomarker, we wouldn't be able to measure the rate of aging even for any drug.
Can I ask a question? Since
You can also take the moderator role.
I thought I was going to be the moderator.
Yeah, go ahead.
Because everything that I do, but even better, like his blog on forums. But I do have a question. I mean, you said there's no treatment for aging yet. Maybe you said that, but anyway. But there's a solution to aging, which is like reproduction, right? I mean, old people, they're producing young people all the time. Even with cloning, an adult cell turns into an animal. So there's like clear and present evidence of immortality as long as the asteroid doesn't hit. So that's why I think we should get to the reprogramming topic. Do you feel that you guys have the tiger by the tail when it comes to sort of some kind of rejuvenation process that would involve reprogramming and resetting these same epigenetic marks that you measure with the biomarkers?
Well, I think that's your new thing. So
Yeah, I'd like to answer. So literally work on that. So what we find is that there's a process of rejuvenation during early development. So when we age, our germline also ages, like moocytes and sperm ages. So the older parents would have older germline, and therefore, like the zygote fertilized egg would be older, a little bit older. But what we find during early development, there's a process of rejuvenation. Approximately in humans, it will be in the third week. So it results in the lowest biological age, we call it ground zero. And that's where we think aging begins, the aging process itself. So that's why we can propagate from generation to generation, simply because we age a little bit in the germline and then rejuvenate, age and rejuvenate. So how exactly this happens, we don't understand. So we see this at the level of epigenetic clocks, we see this probabilistic clocks, we see it at the level of transcriptomic clocks, we see this in mice and humans and frogs. So it seems like it's reproducible, but it survives only our lab. So it would be nice if it would be reproduced by other labs. It may be related to reprogramming that's being studied by those companies like OSK factors, even other factors, because they also express the blastocyst stage. And later, when they express the organism becomes younger. So it would be really, really important to understand the nature of that process. But it's somewhat different process from targeting aging in the adult life. That's a different one. Right? So because in adult life, certain like aging process, they cannot be reversed. In general, I think aging is irreversible itself. Irreversible. So aging, like there's so many factors, so many mutations and interactions that happen. So aging progresses only in one direction, aging process, but not the biological age. Biological age can fluctuate. So we could, biological age can be viewed as the level of damage. Let's say we accumulate damage, and damage may be cleared, can be reduced, for example, or diluted in some way, or removed by activating protective systems, or like by the lymphatic system at night, or renal system, whatever. And so in that sense, you could target biological age. You could reduce it. And we observe this all the time. For example, we have experiments where we connect mice, heterochronic paraviruses. We connect the mice, old and young mouse, and we see the old mouse becomes younger, literally younger at every parameter, and it lives longer as well. Or, for example, we published a paper last year under severe stress. We observed organisms become older. For example, women during pregnancy, in the third trimester, they are older. Postpartum, they become rejuvenated again. I'm not sure rejuvenated, younger, go back to normal age. And under other stress conditions, just an example, like Bryce is here. We tell the student from Stanford spending summer in our lab, and he quantifies changes in biological age during the day. We also observe that we are unable, if we look at from aging perspective, from damage perspective, we can observe. Oh, look here, here. He's a talented postdoc in the lab. He developed this amazing clocks that quantify biological age of organ at organ levels. We profile plasma, and we can tell that in that organism, brain is, let's say, that many years old, kidney that many years old, liver that many years old. And so what does it mean? It means that different organs, they age with different rates. And when we, for example, discuss longevity interventions in general, we're thinking we should target the whole organism, but it's not. Because we are all different, and we age different in different organs. And some interventions might be helpful for somebody who ages, let's say, in the liver, but somebody who ages in the brain more would not be helpful. So I think actually longevity interventions in general are not going to be universal. So they are going to be interventions that will target us in a personalized way. So I'm sorry, I mixed this all so many topics together, but it's difficult topic.
How soon, I guess is the question.
Soon, I don't know. I'll be the first one to do it.
Well, maybe you'll Brian John. To make a joke, but also to continue on this note, I think that the techniques for reprogramming are still in their early days, especially when it comes to human interventions, because if it was easy, we could reprogram cancer, right? Imagine if we wanted to demethylate and try to send it in the senescent state. Still don't have the technology, so I think for reprogramming, we're still in the early days, and we probably will see another decade of small molecules and biologics that will allow us to probe the biology in order to, get us into a specific rejuvenated biological state somewhere in some organ or system. Even sometimes when we don't know how the drug works on aging, we want to first ensure that it works in a disease. Because if you want to do human trials, you need to show that you're addressing a disease condition. So I think for reprogramming, it's going to take time, unless we start clinical trials with technologies that are safe. Most importantly, and I think that some companies went into this direction with ocular diseases and topical applications, but on the systemic level, it's going to be pretty dramatic to do something like that today. So I think we're still stuck with tools we've got like small molecules or biologics. By the way, most pharma still doesn't like even small molecules that have dual purpose, right? So I'll go and try to convince them to buy something that has aging focus. That would be something new. But reprogramming is definitely a promising direction. Currently, it's attracting a lot of investor attention. And hopefully, we're going to see maybe some early steps in topical applications or ocular applications. Would you like to ask another question?
Well,
Let's go off the line.
I do have a question. I mean, like a lot of people, kind of reading what's popping up on social media, that recently, or just the last 6 months, there was all these results that sounded great.
IL-11?
Yes, I'll read what they were, because one thing that would be very concrete would be like, okay, if a mouse that lives 4 years, I guess, on average, maybe a mouse, what if you did something to mice that they could live 8 years? Well, then you would have demonstrated that it lives a lot longer. I mean, it's one thing to do it in a worm. In a mouse, that would be dramatic. So the things that I saw, and I want to hear if Vadim can tell me what they might be as a group, like, is there an acceleration of results? One thing, Rick Klausner, CEO of Altos, claims in a conference that they are extending the life of mice by 25% with reprogramming. We don't know what kind of mice under what conditions, but that's just a conference claim. A claim at the Wisconsin Aging Meeting by Adam Salmon of life extension in marmosets who've been given rapamycin. Some kind of, I don't think we've seen life extension in a primate, a non-human primate, so that would be good, rapamycin. Then a paper on inhibition of IL-11 signaling, 25% life extension in old mice. And lastly, a preprint, rapamycin and trametinib, if I'm saying that right, in combination, extend maximum lifespan of mice by 35%. Something happening with mice and marmosets.
You go first.
This is on the last 6 months.
Yeah, in mice, we have many ways to extend lifespan. We already knew many ways, right? These are just additional ways. We already knew that rapamycin works. We already knew that calorie restriction works. So connected posing, micropose, growth hormone receptor knockout. There's many FGF22 expression. Many approaches. These are additional approaches, but they all fall within the same range, like below 40%, which is already achievable. So a big thing is to go beyond that, because species have the barrier, maximum lifespan, and so we kind of dramatically slow down the aging rate is very, very difficult at the species level. So you could extend lifespan in mice up to 40%. In humans, I'm sure it should be the same, or maybe less, but it should be possible to extend lifespan by same type of approaches like drugs, small molecule drugs, or some other changes. But what would be difficult to go beyond that barrier, that would require radically new approaches, and that's still unclear how to do. But to me, these are none of those highlights you mentioned revolutionary. They fall within what we already know.
Yeah, so I'll just add to that. So the IL-11 work was presented at the IRD 2022, knockout mice, right? But now they did it with an antibody and published a paper that was also presented at the IRD. The rapamycin plus paper was also presented at the IRD. So we know these people very well. I think rapamycin is so established that some people in the field take it. How many of you here take rapamycin? Anybody wants to? Okay, wow. So one confession, right? Congratulations. Anybody else? Oh wow. You.
Oh, no,
All right.
How about you?
I cannot confirm or deny, but I think that if you decide to take it, you probably want to do some resistance training, otherwise lose muscle. And nowadays, a lot of biohackers use rapamycin. So the problem with rapamycin is that it's off patent. So for any pharma company to take it right now and try to do a clinical study for chronic disease, even if they do succeed, and most likely they wouldn't be able to monetize it. So it's kind of a dead drug to pharma, even in combination right now with other generic drugs. For IL-11, we're going to see soon, because Boehringer in-licensed that antibody and took it for fibrosis. They're also doing it for IPF, by the way. So if they are successful and they can get the drug on the market, and even if it's not as strong as, for example, my drug, when they still get an approval, we would be able to see in a clinical setting if that drug is affecting any of the aging clocks. Because of course, my dream would be to, for example, get the IPF drug on the market, and in addition to improving the lung function due to IPF, you would also give patients additional benefit. Imagine that they also get younger. Wow. And actually, most of the IPF patients, what they have in common is their old age. That's kind of one very important feature. Usually, when it's 65, 70, you open up a clinical trial, outcome result with 75, 79 ages. And I think that we are definitely witnessing an explosion of results that are going to motivate people to go and test more interventions in mice. And by the way, those results in mice, like to your point, rapamycin previously increased the lifespan of mice by 15 to 20% in combination of with agarose. So when you see 35%, it's actually a very substantial increase. And when you see 25% with IL-11 antibody, it's also a very substantial increase, right? Of course, they are in cages, sterile environment, so let's see what it does in the wild, but we will see it in humans. So definitely it's very important. And that leads me to another point. So currently we have Novo here in the audience, we have other people who have GLP-1s. Even GLP-1 has not been tested in mice for a lifespan intervention. We don't know what it does longitudinally to mice. Why didn't you do those studies? Right? So my challenge to the pharmaceutical industry is that when you are doing, let's say, your 28-day GLP tox, and you want to see if the drug has some effects for chronic administration, why not do a lifespan study in aged mice, run it for 9 months, and then see, first of all, if there are any longitudinal side effects for chronic administration, and B, if it actually increases their lifespan. So for that, we of course need to go and to convince the FDA not to punish you if you see some nasty effects, right? And for example, if it decreases the lifespan, it's still, so even for me, I'm like super dedicated to longevity. Doing a lifespan study on one of my drugs, you don't even know the dose, right? But I think if those lifespan studies were available right now from CROs for, let's say, $50,000, $60,000 and 9 months, you would see an explosion of those studies, and we would see a headline like what you just described every day.
Right. Well, people are treating it like a life extension drug. I mean, I was just on social media, somebody posted one of the charts from a paper, and it's like a survival curve for humans. It looked just like the mouse one, but they're on these inhibitors. It has so many benefits for the heart, this, that, or the other. As you point out, it's also this huge uncontrolled experiment. So I think history tells us that 1,000 people take it, but now it is, I don't know exactly the numbers, but it is in the tens of millions of Americans that are on this drug. I mean, it is like a large part of the population. So if there's something bad that's rare, we're going to find out.
Yeah.
And what do we think? Is it more likely that it's something bad and rare, or something good and unexpected like a longevity?
Most likely,
do you think about this drug, this class of drugs?
Well, this was no drugs being studied, so I don't know, but the data idea quite promising. Actually, it make a real longevity drug. So we just need more data on that, but it is possible.
Agreed 100%. I think that GLP-1s right now, if I were a little bit kind of bigger, a little bit more fat tissue, I would probably be on it right now. And I probably should anyway. But then problems with GLP-1s is that we actually know from clinical trials that up to 40% of the weight loss is muscle loss. So you are going to lose something, right? And you don't really want to lose muscle. So right now we're actually trying to figure out how to combo it with all kinds of drugs like apelin receptor inhibitors and others. A lot of people are looking at artinic because of the recent study from the University of Copenhagen showing that knockout mice don't gain fat and maintain muscle or gain muscle on high-fat, high-sugar diet. But I definitely think that GLP-1s for people who are, let's say, a little bit overweight, very good idea to go on those drugs, but plus also start exercising, specifically resistance training. Those kind of dual benefit. I don't want to lose muscles. There is always a trade-off. My problem with this drug is that it hasn't been tested in mice for lifespan. Why? And my problem with most other drugs is that why they have not been tested for lifespan, especially if you're addressing a chronic disease. So I think that once we see more lifespan tests for any drugs, once it becomes more mainstream, we will see an explosion of experiments where all kinds of drugs extend lifespan in mice.
Actually, the lifespan studies in mice are not that typical. So especially for academic labs funded by NIA, they have a really nice program where we could receive all the mice, so not many mice, but some mice every month. So they're utilizing. So we actually have many studies on aged mice currently in the lab. But I also wanted to stress this point of like asynchrony in aging across different organs. So we're still talking about drugs like for everybody. So I really not really sure. Let me give you like a really simple example, just from Luker's work here, sitting here. So let's say he has an intestinal aging. So he has a clock based on human plasma where he can predict the age, intestinal age. And he has another clock from human plasma that predicts the age of the artery. And he finds that like alcohol drinking, we know it's not good, right? But apparently for the artery, at least for arterial clot that he develops, it is beneficial. So it kind of rejuvenates the artery while accelerates intestinal aging. On the other hand, sugars, which are also generally bad, have the reverse effect. So I wonder if somebody might age more in the artery. They really should not eat sugars, but maybe wine from time to time is okay. It's all speculation, right? We don't know for sure. But somebody ages, more in the intestinal aging, it should be the opposite. So how do we know? Let's say if we GLP-1, I don't know, metformin, rapamycin, whatever, we need to know who can benefit. Not everybody will benefit from any of them. And this is something which has not been addressed at all. So I think maybe in the future it will be addressed, but we need to do it.
Very good point. I actually think that in addition to alcohol, I think like Cialis or Viagra would probably also have very similar effect because it should improve the elasticity of the blood vessels, right? In theory, nobody has done this study, but there is a lot of evidence now in the literature. So people should do those clocks as well. And Vladimir, my question to you: what is aging?
Well, I do aging in terms of accumulation of damage. So, and damage not in the form of just like reactive function species or whatever, some kind of telomere shortening, more global. So when we live, we have processes that run in the cell, like genes and metabolites, proteins. They do whatever they do, but as they do, they also produce negative consequences. It could be a mutation or mutation, could be some kind of wrong metabolite, could be modification in a protein, many slightly different level of a hormone or gene expressed, and so on. And those changes, there are many. They come from the totality of processes that we have, and it's really organism is unable to deal with the entirety of that damage. It can deal with some of the damage, but not with the entirety. So it grows over time, unavoidably. If you have an organism like mammal, with like fully differentiated non-renewable cells like neurons or cardiomyocyte, eyes, skeletal. That's it. We are done. You cannot stop it, aging. It continues. But we can slow down. We can still target biological age in some way to reverse it under some conditions. So we could still make a difference. I don't think we can stop the aging process. So that in essence, it's my model may be wrong. I don't know, but because there's no consensus.
How would you cure it?
That's what I said. I mean, you cannot cure it. You can slow down and you can partially reverse in some systems by targeting biological age.
But what about with tools available today? What do you think is the most promising direction?
Okay, the most immediate to me is still small molecules, as you said, because they are safe. It's easy to develop. Except, I guess maybe I disagree with the previous panel here, because you guys all have targets, right? So you need targets. We don't care about targets. Our target is the whole system. So we ask the question how a particular compound changes the system. The system in terms of like biological age, whether it targets a particular gene or particular protein or 10 proteins or 100 proteins, doesn't matter. As long as it changes the system in the direction of longevity. And that's our target. So we view aging as a systemic process and we address it systemically. And it's somewhat the contradiction with the traditional pharma model, but I think in the future it will still come if we want to target the aging process. I'm not saying targets are bad. Targets are also good. They can be targeted and some success may be achieved. It's just, I think, a better success is going to be targeting aging system. That's all.
Agreed 100%. Antonio, would you like to ask a question?
I thought you were going to ask me.
Okay, well,
but I didn't know.
Well, in this case, I will ask you the question, and then we will conclude the panel and ask people to ask questions. My last question would be, what do you think is hot in biotech right now? Outside aging, inside aging, what is the hottest trend?
I was talking to the business development person from Sanofi, and I said it might be like if something matters, if it's worth money, I stop covering it. So I was working on like an emerging thing, a speculative thing. My work keeps coming back to developmental biology, to the embryo. I mean, lately they've been sort of making embryos from stem cells called synthetic embryos. That is amazing, to create a life form of sorts from a single stem cell. So to me, that is kind of where the quickening is going on. It's irrelevant to business. It's barely legal to do.
Hugely irrelevant, but hugely legal, by the way, depending on how you.
Yeah. I mean, they're not
Because
quite.
there's no conception.
Yeah.
They make embryos from stem cells. So therefore there's no restriction because there's no conception.
Exactly.
Yeah.
Especially if you use like a mouse Excel.
So there's a whole set of technologies to do with reproduction, which are super speculative and people are against. And then, there's this thing with the editing of the germline, like the CRISPR babies. I mean, human population genetics has definitely found, I think, so if you agree, it definitely has found some, even single letter genetic changes between people, which are massively beneficial. No, you never get Alzheimer's if you have one of them, on the right genetic background. So people have dropped this discussion about the germline editing, the guy trying to prison, but I'm like, sorry, where's the plan? Like, this is like the biggest thing that ever happened, that we could kind of control evolution and install beneficial variants on Earth. So, that discussion has stopped, but I think it's interesting to restart it. So I look around for people like George Church and others that are willing to talk about it. So I'm sort of looking for the people who are, want to improve the species, so to speak.
What is the hottest thing in biotech? Any ideas?
I don't know. I mean, there's nothing that is really, really hard at the moment. I mean, things are improving in the aging field. Like in mice, we have many interventions that extend lifespan that you mentioned some. So I think some of them will be tested in humans. Human lifespan will be extended to some degree, maybe 5%, maybe 10% in a somewhat near future. So I think it will be a really good development. Later, I guess more radical approaches could be tried, like reprogramming, but we don't really know whether it would work or not. Or maybe like organ replacement, very promising. But again, so it's not in the near future, presumably. So it's hard to
So
say.
you have a conservative approach.
No, I'm very forward-looking. Not conservative at all, but I think we need to be realistic.
Well, thank you. I think that the hottest thing in aging research for drug discovery currently is the TNIC program. And PhD 1-2 systemic, hopefully. We'll see. But now let's open the floor for questions. Here you've got the huge center of gravity in aging and the purist who really understands aging and talks about the reality of life. And you've got exponentialist who looks at radical technologies, right? And is curious about things that are outside of norm and at the cutting edge, right? So let's go.
Very cool panel, thank you. Interesting. So as an outsider into aging research or biotech activity, I recently, I was just this weekend in Barcelona, where ESMO Clinical Coach Conference happened, and I met some executive from Grifols. The Grifols company was in plasma products, which are in phase 2 trials with anti-aging and prevention of neurodegenerative diseases, Alzheimer. This plasma reflective. And now also they're getting into anti-aging research. So I'm wondering what both of you think about this and in prediction for such approach. Of course, it's not as cool as some other approach you mentioned, but given what you said, both about the organ different sensitivity of organs to or speed of aging in different patients, how do you see that? Any predictions for this approach? And also, just should I do something?
Could you clarify, is it injection of plasma?
It's just
plasma
blood
fraction?
transfusion, which has been used for many years. Simple blood transfusion.
From like younger blood?
Younger blood, this is yours.
Great question from the senior BD person from Astellas. So exponential.
Well, I was trying to write the notes because it sounds like a good story. What was the name of the company?
Grifols.
Grifols.
It's been around over 100 years.
Yeah, but they're pretty ancient guys, right? So I think they bought something from
They
California.
bought something that is on legend.
Yeah.
I just checked two things on Google appears their patent on the engine and the company they bought something with A.
Well, journalists love this topic because, of course, it has like two mice sewn together. It's very grisly. And then you watch this. Then there could also be a blood donor. There was a show on TV called Silicon Valley. You have your young blood boy. But interestingly, I think Benny will have the comment. Most recently, what I've heard is that people are trying to. Okay, so blood has a lot of stuff in it. So what is it in the blood that would be having a beneficial effect? And so I think that they have found that actually if you just dilute someone's plasma, that is the beneficial effect. Like I think it's salt water and albumin, maybe.
Yeah.
So that's what people are getting. He's nodding with great, like he really knows about it. People are going to get. That's what people are going to get because it's also like generally recognized as safe. I don't think people want to do it every day, but it's a dilution of your plasma. I think people, you can go to a clinic in San Francisco and they will do this for you.
Okay, so we have experiments on mice on parabiosis. We connect young mice and old mice for 1 month or 3 months. 3 months is like 10% of their life. It's really long. And you find that the old mice become younger. They also live longer. If you do it for 1 month, you barely see the effect in mice. I mean, we see them in older mice a little bit younger. We haven't checked the lifespan after 1 month parabiosis. But it's not just exchange of the blood. When the mice are connected, old mice also have access to the younger organs. let's say they want to remove some damage from the kidney, the liver, and so on. And it's a continuous exchange of drug. And the effect is still relatively minor. I can't imagine, I know there are companies and people publish papers. I just don't understand. Another, if it's just injection one-time blood, how can it have an effect? I'm really skeptical. Also, dilution of blood. It's a promising approach, but I don't see strong evidence in this case as well. So I think more data needed. Maybe if the blood, like young blood, is injected and checked at a certain time, because you inject the blood and you can monitor biological age, at some point you might find, okay, maybe it's decreased because the organ is activated protective resources, and then you see the effect, yeah? And then you publish a paper. But you need to continuously look, or like you inject and maybe follow like lifespan, for example, like in old mice, and then it will be more convincing. So at this point, I would say there is not enough data. Okay.
All right, so there's a question here. I will be the microphone guy.
Okay. I'm very interested in the different aging of organs, and I'm curious if you've identified organs or organ systems that are more impactful on overall aging, or also organs that are correlated with each other. Because you talked about them having different ones, but could you, for example, check the aging of your skin, and that's a signal of your gut aging? Or, have you seen any patterns there yet?
Well, I didn't even know the answer to this question.
May I ask this person? I mean, just to give him a chance to speak.
Sure, yeah, I mean,
Just introduce yourself.
Yeah, I'm Rutger. I'm also in Berlin's lab, and I work on the organ-specific aging based on proteomics in the blood. Actually, they all tend to correlate with each other overall. So somebody who is older in one organ tends to be older in the other organs. Could be, I should check again. I think it could be that some clustered it together, but it was not super clear. There were no clear clusters.
And in some other studies, we also have not really found like a key organ. I mean, like for example, by organ replacement, but it would be interesting to replace a brain and check that. Quite hard to do.
I don't think it's replaceable yet.
Well,
you covered that.
Oh yeah. No, but the companies, including the reprogramming companies, I mean, if the intervention is a genetic intervention, a lot of times, what organ are they looking at? They're looking at the blood system, bone marrow, because you get the cells in or out, T cells in or out. So just in a practical way, the answer is the liquid organ that you can move around.
The most easily accessible.
Yeah, and also it touches everything. I don't know what the scientific answer to your question is, but the practical answer is that there's a lot of these efforts starting with T cells.
Yeah, from a logical perspective, I think that lung, liver, and other very fast regenerating organs that also accumulate a lot of fibrosis, they would be the first to go. And it's kind of counterintuitive because at the same time they are regenerating, but also they're accumulating fibrosis. That's one of the reasons why we decided to go to teenage, because it's an anti-fibrotic, plus the score is very high in many chronic processes. But I would like to have the opportunity for you to interact with the speakers a little bit and interact with yourself, because many of the people here are like somebody. Most of the people here are high-profile people from pharma, from the biotech community, and I would like to thank you for coming here. And we are open now, so Insilico Boston is where to go. And we are focused on AI, on drug discovery, and on aging research. Because aging unites all of us. It would be great for us to discover interventions that target both disease and aging in the most commercially tractable, ethical, and legal way as well. Because because you don't want to do things that are outside the norm too dramatically. So please interact with each other where we have, so outside where we have a patio, we have servers providing food, drinks. So please do enjoy yourself and interact with the speakers. But we wanted to set the tone. Business development, pharma, and aging research. And out of the box. So high tech. So thank you very much, and let's give a warm round of applause. If you are an investor and if Vladimir starts a company. Well, thank you.